Hello everyone. Today, I'll present my research on CAPTAN, a widely used fungicide, focusing on its efficacy, ecological impacts, and sustainable use in agriculture. By the end, you'll understand CAPTAN's benefits, risks, and how to balance its use for sustainable farming. Here's what we'll cover. First, an introduction to CAPTAN, its history and uses. Then we'll dive into 3 key studies: its compatibility with biocontrol fungi, efficacy against rice diseases, and toxicity to honeybees. Finally, we'll discuss conclusions and future directions. Broad-spectrum fungicide developed in 1951 by Chevron for World War II rubber protection. It disrupts fungal cell metabolism, making it effective against diseases like apple scab and botrytis. In Myanmar, CAPTAN boosted mango yields by 15% from 2018 to 2022. CAPTAN was revolutionary in 1954 as orthocidar but later flagged as a probable human carcinogen, US EPA 1989, and banned in the EU in 2019 due to ecological risks. Its formula is C9H8Cl3NO2S. It degrades quickly in sunlight, 2 to 3 days, but persists longer in soil, 3 to 10 days, raising concerns about residue. Farmers use CAPTAN at 1.5 to 4.0 kg/ha every 7 to 14 days. However, it's toxic to aquatic life and classified as carcinogenic. PPE is mandatory in the U.S. This 2024 study tested CAPTAN's compatibility with 4 trichoderma species, beneficial fungi used for biocontrol. The goal? To see if they can coexist in IPM strategies. These are the good guys, trichoderma fungi. They fight pathogens like fusarium and botrytis by parasitizing them, competing for nutrients, and boosting plant immunity. Their detox genes, ABC transporters, help them tolerate low-dose fungicides. Researchers used a poison food technique, exposing trichoderma to CAPTAN at 450 to 1,350 mg/L. CAPTAN was 79.87% compatible, best for T. harzianum. Mancozeb was moderate, 72%, while chlorothalonil was toxic, 15%. Figure 1 shows CAPTAN's dose-dependent inhibition of trichoderma growth. Even at high doses, some species thrived. PCA analysis, Figure 2, confirmed 93.1% variance. Proof that trichoderma responses depend on both species and fungicide type. Why does trichoderma tolerate CAPTAN? ABC transporters detoxify it. These fungi also attack pathogens directly and boost plant immunity. Nature's pest control, enhanced by science. Mancozeb disrupts cell membranes. CAPTAN blocks enzymes. But CAPTAN's synergy with T. asperellum against anthracnose, see footnote, hints at smart combos. Chlorothalonil? Just avoid it. Farmers should pair CAPTAN with T. harzianum for sustainable disease control. Rice loses 50% yield to sheath blight. This study pits CAPTAN against mancozeb. Meet Rhizoctonia solani, the culprit behind sheath blight. Those lesions? They decimate yields. Non-systemic fungicides like CAPTAN are safer for food chains but need proof of efficacy. Poison food technique. Fungicides mixed into agar. Field trials mirrored real-world conditions. Mycelial inhibition is measured against untreated controls. Visual proof. From right. Increasing doses stop Rhizoctonia dead. CAPTAN at 1000 g/ml? 100% inhibition. But field results, next slide, tell the real story. Mancozeb wins, but CAPTAN is close. 93.8% inhibition. Field data shows both reduce disease and boost yields by 20%. Copper oxychloride? Forget it. CAPTAN and mancozeb are top choices for sheath blight, but copper oxychloride failed. This shows the need for targeted fungicide selection. Bees are vital pollinators. This 2024 study exposed larvae to CAPTAN and thiamethoxam, a neonicotinoid. The results? Alarming synergism at high doses. Workers run the hive. Drones die after mating. Why care? Bees pollinate 1/3 of our food. Captain's toxicity risks ripple through ecosystems. Larvae-fed pesticide-laced diets. Colonies monitored for a year. Dosing 100 ppb. Captain mimics field exposure. Real world but controlled. Royal jelly, glucose, fructose, be gourmet. But add Captain and survival plummets. Day 4 is critical. That's when pesticides hit hardest. Captain alone caused 80 to 90% larval mortality. With thiamethoxam, mortality spiked to 210%. Yet field colonies survived, likely due to social buffering. See the red line? Captain plus thiamethoxam is lethal, but green control versus blue Captain low shows colonies adapt. Still, regulators should heed this warning. Asterisk. Larvae are most vulnerable. Prepupal stages resist better, but synergism is deadly. This demands rethinking safe pesticide cocktails. Field colonies compensated, brood and pollen stores stayed stable. But solitary bees lack this resilience. Captain's risks aren't one-size-fits-all. After 12 months, colonies survived, but plus marks censored data. Some died. Chronic effects? Unknown. We need longer studies. Captain's Be Safe label needs reevaluation. While colonies compensate, wild pollinators, e.g. solitary bees, may not. Avoid spraying during bloom and study chronic exposure effects. In labs, Captain is toxic. In fields, colonies compensate, but this isn't a free pass. We must study wild pollinators and chronic exposure. Recap. Captain shines with trichoderma and rice, but bees pay the price. The verdict? Use it strategically. Low doses avoid bloom periods and pair with biocontrol. Captain is a tool, not a cure-all. Integrate it with IPM, protect pollinators, and push for smarter regulations. Sustainability isn't a buzzword. It's survival. These are commercial Captain formulations. Note the 50% WP, wettable powder label. Always follow dosage guidelines to minimize risks. All studies cited here are peer-reviewed. Dive deeper, especially the 2024 bee toxicity paper. Science evolves, and so should our practices. Thank you. Questions? Let's discuss how to balance crop protection and ecological health. The floor is yours.